Imagine shrinking down to the size of an atom, a world so small that it defies our everyday understanding of reality. Embarking on a journey into the bustling metropolis of a single cell, we find ourselves in a realm teeming with activity and life. Within this microscopic universe, intricate machines whir and hum, carrying out the symphony of life. These tiny structures are constantly at work, ensuring that every function necessary for life is performed with precision. These are proteins, the workhorses of our cells, orchestrating every biological process from transporting oxygen to fighting off infections. They are the unsung heroes, tirelessly working behind the scenes. Understanding these protein machines is key to unlocking the secrets of life itself. By studying their structures and functions, scientists can uncover the mysteries of how life operates at the most fundamental level. By deciphering their intricate structures and interactions, we gain insights into the fundamental processes that govern our health and well-being. This knowledge is crucial for advancing medical science and improving human health. This knowledge empowers us to develop life-saving drugs and therapies for a wide range of diseases. From cancer to genetic disorders, understanding proteins opens the door to innovative treatments. The study of proteins is a journey into the very essence of life, a quest to understand the molecular machinery that makes us who we are. It is a field that holds endless possibilities for discovery and innovation, a quest to understand the molecular machinery that makes us who we are. Each discovery brings us closer to comprehending the complexity of life itself. It's a realm where cutting-edge technologies are constantly pushing the boundaries of our knowledge, allowing us to see and understand things that were once beyond our imagination, revealing the beauty and complexity of the microscopic universe within. This hidden world is a testament to the marvels of nature and the endless pursuit of scientific discovery. Proteins are the building blocks of life, and their structures dictate their functions. These complex molecules are involved in nearly every process within our cells, from catalyzing metabolic reactions to replicating DNA. Without proteins, life as we know it would not exist. Imagine a protein as a complex origami creation, carefully folded into a unique three-dimensional shape. This folding process is not random. It follows a precise sequence dictated by the protein's amino acid chain. Just like a master origami artist, the cell ensures that each protein folds correctly to perform its specific function. This intricate shape determines how the protein interacts with other molecules, allowing it to perform its specific job. For instance, enzymes, which are proteins, have active sites that bind to substrates with high specificity, facilitating biochemical reactions essential for life. For example, the hemoglobin protein in our red blood cells has a unique shape that allows it to bind to oxygen in our lungs and transport it throughout our body. This efficient oxygen transport system is vital for our survival, as it ensures that all our tissues receive the oxygen they need to function properly. A slight alteration in this shape can disrupt this process, leading to diseases like sickle cell anemia. In sickle cell anemia, a single amino acid change causes hemoglobin to form abnormal shapes, leading to red blood cells that are rigid and sickle-shaped. These malformed cells can block blood flow, causing pain and organ damage. Understanding protein structures is crucial for developing new drugs and therapies. By studying the detailed architecture of proteins, scientists can identify potential targets for drug development. This knowledge allows for the creation of medications that can precisely interact with these targets, improving treatment outcomes. By knowing the precise shape of a target protein, scientists can design drugs that bind to it with high specificity, like a key fitting into a lock. This specificity is crucial for the effectiveness of the drug, as it ensures that the medication will interact only with the intended protein, reducing the likelihood of side effects. This targeted approach minimizes side effects and increases the effectiveness of treatments. Patients benefit from more precise therapies that address the root cause of their conditions, leading to better health outcomes and improved quality of life. From fighting infectious diseases to developing personalized medicine, the study of protein structures holds immense promise for improving human health and well-being. As our understanding of these complex molecules deepens, we can look forward to a future where diseases are more effectively treated and health is more easily maintained. Cryo-Electron Microscopy, or Cryo-EM, is a revolutionary technique that has transformed our ability to visualize protein structures in exquisite detail. This method has become a game-changer in the field of structural biology, providing insights that were previously unattainable. Imagine taking a snapshot of a single protein molecule frozen in time at extremely low temperatures. This snapshot captures the protein in its natural state, without the need for crystallization or other modifications that might alter its structure. That's essentially what Cryo-EM does. It allows scientists to observe proteins as they exist in their native environments, providing a clearer picture of their true forms and functions. Here's how it works. A solution containing the protein of interest is rapidly frozen, trapping the protein molecules in a thin layer of ice. This rapid freezing process is crucial because it prevents the formation of ice crystals that could damage the protein structure. This ice is then bombarded with a beam of electrons, which pass through the sample and hit a detector. The electrons scatter as they pass through the protein, creating a pattern that can be analyzed. By analyzing the patterns created by the scattered electrons, scientists can reconstruct a three-dimensional model of the protein structure. This 3D model provides invaluable information about the protein's shape, function, and interactions with other molecules. Cryo-EM offers several advantages over traditional techniques for determining protein structures. Unlike X-ray crystallography, Cryo-EM does not require the protein to be crystallized, which can be a significant hurdle for many proteins. It can be used to study proteins that are difficult to crystallize, a prerequisite for X-ray crystallography, another structure determination method. This makes Cryo-EM particularly valuable for studying membrane proteins and large protein complexes. Cryo-EM also allows scientists to visualize proteins in their native states, providing a more accurate representation of their structures and dynamics. This native state visualization is crucial for understanding how proteins function in living organisms. This powerful technique has opened up new avenues for understanding protein function and has already led to breakthroughs in drug discovery and disease research. By revealing the intricate details of protein structures, Cryo-EM is helping scientists develop new treatments for a wide range of diseases, from cancer to neurodegenerative disorders. While Cryo-EM provides a detailed snapshot of individual protein structures, Cross-Linking Mass Spectrometry, XLMS, sheds light on how proteins interact with each other. Imagine having a map that shows not only the locations of different buildings in a city, but also the roads and bridges that connect them. That's what XLMS does for protein interactions. In this technique, proteins are treated with special chemical cross-linkers that act like molecular bridges, linking together amino acids that are close to each other in space. These cross-linked proteins are then digested into smaller fragments and analyzed using mass spectrometry, a technique that identifies molecules based on their mass-to-charge ratio. By analyzing the cross-linked peptides, scientists can identify which parts of the proteins are interacting with each other, providing valuable insights into their functional mechanisms. XLMS complements Cryo-EM by providing information about protein-protein interactions, adding another layer of understanding to the complex world of cellular machinery.